Every human life starts as a single diploid cell. That one cell divides into 2, then 4, 8, and 16, packing more and more cells into the exact same volume, until it eventually scales up into an adult structure of roughly 37 trillion cells. This growth presents a structural paradox. Every single one of those 37 trillion cells contains the exact same genetic code, yet a neuron in your brain, a muscle fiber in your heart, and a gut epithelial cell perform vastly different tasks. A cell takes on the specific structure, function, and biochemistry of a specialized tissue through a process called differentiation. Before a cell can differentiate, it has to undergo determination. This is the moment a cell makes an irreversible commitment to a specific developmental lineage. Embryogenesis proceeds as a deterministic sequence of physical spatial sorting and selective gene transcription. To map how this works, we will track the morphological cascade that leads to the formation of the nervous system, analyze primary microRNA data that dictate cell fate, and examine the highly specialized circulatory shunts that keep a fetus alive. This process relies on a strict spatial logic, where a cell's physical location within the embryo dictates its ultimate biological destiny. The earliest stage of spatial sorting happens during blastulation. The dividing cells segregate into 2 distinct groups, the inner cell mass, which will become the actual organism, and the surrounding trophoblast, which will form the placenta. Once implanted in the uterus, that inner cell mass undergoes gastrulation, a process that generates 3 distinct primary germ layers. This diagram illustrates gastrulation. An invagination called the archenteron pushes deep into the blastocele cavity. This physical movement establishes the core axis of the human body and resolves into a 3-layered gastrula. The outermost layer is the blue ectoderm. It gives rise to the integument, your skin, hair, and nails, along with the lens of the eye and the entire nervous system. The middle layer is the red mesoderm. It develops into the musculoskeletal, circulatory, and excretory systems. Finally, the innermost layer, the yellow endoderm, forms the epithelial linings of the digestive and respiratory tracts. These origin pathways create specific anatomical quirks. For example, the adrenal cortex derives from the mesoderm, but because the adrenal medulla contains nervous tissue, it derives from the ectoderm. This initial spatial sorting during gastrulation acts as an irreversible partition. It locks previously undifferentiated cells onto one of 3 strict developmental highways. Since the nervous system derives from the ectoderm, it starts on the outermost surface of the embryo. This raises a mechanical question. How does surface tissue end up forming the internal central nervous system? The answer is induction. One group of organizing cells secretes chemical signals, called morphogens, to dictate the fate of adjacent responder cells. This animation shows the start of neurulation. A rod of mesodermal cells called the notochord forms along the long axis. The notochord acts as the organizer, releasing a chemical gradient that induces the overlying blue ectoderm to fold inward. The morphogens cause neural folds to rise and surround a central neural groove. As the folds grow toward each other, they eventually fuse to form a closed neural tube. This newly formed neural tube pulls the surface tissue inside and eventually becomes the central nervous system. Meanwhile, the cells at the very tip of each fold, the neural crest cells, break away and migrate outward through the body. They will form the peripheral nervous system. Induction serves as a critical biological failsafe. By relying on short-range chemical diffusion, the embryo ensures that tissues which must wire together in the adult body develop in strict proximity. But how do those cells know which part of the nervous system to become? At the molecular level, how does a neuroectoderm cell decide whether to turn into a neural tube epithelial cell or a migrating neural crest cell? To find out, researchers isolated a specific molecule called microRNA 29B. They set up 3 test conditions: a normal control group, a microRNA sponge group engineered to competitively bind and inhibit the microRNA, and a group engineered to overexpress it. This bar graph displays the relative expression levels of ZFP521, a marker gene for neural tube epithelial differentiation. When comparing the 3 conditions, a clear pattern emerges. Overexpressing microRNA 29B drastically spikes neural tube differentiation compared to the control. Conversely, the sponge condition, which strips the microRNA out of the environment, suppresses it. However, if we look at the alternative cellular fate, becoming a neural crest cell, the data flips. This chart shows the relative expression levels of neural crest cell markers, specifically P75 and SNAIL2, across the exact same 3 test conditions. Here, the sponge condition, the complete removal of microRNA 29b, significantly increases the expression of these neural crest markers. And as expected, forcefully overexpressing microRNA 29b actively suppresses neural crest development. The data proves that microRNA 29b functions as a binary switch. Its presence forces neural tube formation while actively blocking the neural crest alternative. While cellular fate is dictated by local molecules, the developing fetus must also solve a massive systemic problem. Prior to birth, the fetal lungs and liver are completely non-functional and highly sensitive to blood pressure. Because these organs cannot process air or waste, gas exchange and detoxification are entirely outsourced to the maternal placenta through passive diffusion. To ensure oxygen flows in the correct direction across the placental barrier, fetal red blood cells rely on fetal hemoglobin. This specific protein has a higher affinity for oxygen than adult hemoglobin, allowing the fetus to actively strip oxygen from the maternal blood supply. Because oxygenation occurs at the placenta instead of the lungs, the standard definitions of blood vessels flip. The umbilical vein carries highly oxygenated blood toward the fetus, while the 2 umbilical arteries carry deoxygenated waste away. This schematic traces the path of fetal circulation. To keep blood actively routed away from the underdeveloped lungs and liver, the fetus constructs 3 temporary anatomical shunts. The first is the ductus venosus. It takes the bright red oxygenated blood returning from the umbilical vein and shunts it directly into the inferior vena cava, entirely bypassing the liver. Once blood reaches the heart, it hits the foramen ovale. Because the right side of the fetal heart is at a higher pressure than the left, this one-way valve pushes blood directly from the right atrium into the left atrium, completely skipping the pulmonary circuit. Finally, the ductus arteriosus catches any residual blood that happens to make it into the pulmonary artery, immediately shunting it down into the aorta. The fetal cardiovascular system is a purpose-built temporary hydraulic machine engineered to perfectly exploit maternal resources. Embryogenesis relies on a precise chain of events where localized chemical communication scales up into complex systemic engineering. Localized morphogen gradients and specific microRNA expression profiles force early pluripotent cells into strict primary germ layers. Those germ layers construct a temporary physiological state, utilizing fetal hemoglobin and specialized circulatory bypasses to survive in utero. This temporary state only ends when the peptide hormone oxytocin and local prostaglandins trigger the rhythmic uterine contractions of birth. The physical act of birth alters the organism's entire biology. The moment the umbilical cord is severed, the fetus loses its connection to the placenta, causing a massive, immediate shift in systemic vascular resistance. This sudden reversal of internal pressure forces the foramen ovale and the ductus arteriosus to slam shut. For the first time, blood is routed entirely to the newly inflated lungs. The deterministic cascade that began with a single cell's cleavage culminates in the precise moment a temporary hydraulic bypass closes and autonomous human life begins.